Hello. Very good evening to the student who have joined. Can you please let me know if my voice is audible to you? Kindly let me know through the chat box if my voice is audible, and also please let me know if you can see the presentation. Hello, am I audible? Kindly let me know if I am audible and if you can see the presentation. Okay. Thank you very much for confirming. We will be starting our session at 6 p.m. Till then, please wait. If you have any doubt or query from a previous class, you can let me know now. In our last class, we have discussed about the computer in medicinal chemistry, antibacterial agents, and combinatorial chemistry. So, if you have any doubt or query from the whatever we have learned in last class, please you can let me know now. Okay. Otherwise, please wait till it's 6 p.m. Then we will start our session. Okay, so it's 6 p.m. So I think I should start the class. So very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Medicinal Chemistry, and today it is our week 11 live session. So before starting the session, like every class, I would like to point out few things that there will be problem solving session every week, and these classes are scheduled on Monday from 6 to 8 p.m. Okay, so we have this medicinal chemistry course. In our medicinal chemistry course, actually, it is a 12-week course. So for each week, we are having each problem solving session, right? So today it is the 11th problem solving session. On the next Monday, we will be having our last regular problem solving session. And after the day after that means Tuesday, I think it is 14th of April, we will be having our summary session. That summary session will be the last class from our this course. Okay, so all the details will be communicated to you via email. So the classes are live on Google Meet platform, and this is the link for the class. The link will remain same for our upcoming class. Means our last regular class and also the summary class. The link will be same. Okay. So if any network disruption happen from my side, do not leave the session. Okay, I will try to join as soon as possible. During the session, you can post your doubts and query through the chat box. I am recording the session, and this recording will be shared to you, and it will be uploaded after the session, or by tomorrow I will surely upload the recording. Okay, and because I'm recording the session, I have muted your microphones and cameras so that I can record the session without any problem. But the chat box is always open, and the chat box we will use for the question answer session in our class also. And also you can let me know your doubts, your query, whatever it is, you can let me know through the chat box. Okay, if doubt persists after class, you can drop an email to me. I will answer your queries through the email also. This presentation which I am showing today and the class recording, it will be shared to you in the SWAM portal. So where you can find it, like every day I will just show you one more time. You have to go to the SWAM portal. Okay, so this is the SWAM portal of your course of medicinal chemistry. This is the announcement page. So on this page, on the left panel, you will find in under the course outline, there is a thing which is called as problem solving session January 2026. If you click on the problem solving session recording, you will be taken to a page which looks like this. On clicking this link, you will be redirected to a page which looks like this. This Google Sheet contain all the YouTube link for our previous class recordings. Okay. Till now we have 10 classes already, so all the links of the class recordings, all the YouTube links are given here, and also there is a presentation folder link. This is a Google Drive link. If you click on that Google Drive, you will find all the presentation, whatever we have done till now. Till now, they had the at the 10 presentations for our previous 10 classes. You can download the presentation from here and you can go through it. Okay. So all these things are available on the SWAM portal. You can find it out from there. And also, one more thing, I think your examinations are also, I mean, declared. So you can find all the details regarding this examination also in the this SWAM portal. So you will find for the medicinal chemistry, it is a CBT or computer-based test, and the exam format is available here. For computer-based test, the questions will be entered through the computer. Type of question may include multiple choice question, fill in the blanks, and is essay type answers. Okay, so these will be type of examination and mode of examination. So all the details are given here. You can go through it. So I think I have mentioned all the details which you need to know for our class. So today, as I have mentioned, it is our week 11 live session. So in our last class, what we have done? So till now, we have learned the basics of the medicinal chemistry. Okay, and we have seen why drug discovery is important. We have both gone through the pharmacokinetics, pharmacodynamics, all the details of the those things we have understood. We have understand the phase 1, phase 2 metabolism. Also, we have discussed various drug designing techniques like the methods of the like the extension or the this kind of things we have already discussed, right? So in our last class, as I clearly remember, we have discussed a very important topic, that is the antibacterial agent. So I have told you that we were going to start regarding various therapeutic agents and the diseases from the last week. So in the last class, we have discussed very in detail the antibacterial agents, the classes of antibacterial agents from penicillin, then cephalosporin, and then we have discussed about the quinolone, macrolides, and oxalidinones, and all these things we have discussed in our last class, right? Also, we have discussed about the computers, importance of computers in medicinal chemistry and combinatorial chemistry. So these are things already we have learned in our last class. So what is the course content of today's class? So today we are going to understand various topics. So mainly three different therapeutic topics we are going to discuss today. One is the antiviral agents. Second, we are going to discuss about the anti-cancer agent, and then we are going to discuss about cholinergic and anti-ulcer agents. Okay, so three different topics and three different therapeutics area that we are going to discuss today. So this is a very important thing, and to understand, if you have any doubt or any query at any point of the class, please let me know through the chat box. Okay, the chat box is always open, and I am monitoring the chat box every now and then. So if you have any doubt or any problem, please let me know through the chat box. Okay. So like every day, we will be having 20 MCQ question related to these topics. Each and every MCQ question will have four different options. After showing you a question, I will wait for some time and you have to let me know A, B, C or D, which of the following will be the correct answer. Okay. And then I will show you the answer and we will discuss that particular topic in detail. Okay. So let us start with the first question of today's session. The first question, it says that which of the following statements is true regarding virus? Your options are: number A, viruses are not living organisms. Number B, viruses grow and reproduce outside the host cell. Number C, tuberculosis is a viral disease. Number D, viruses are giant compared to bacteria. Which of the following statements is true regarding the virus? Again, I will repeat. Number A, viruses are not living organisms. Number B, viruses grow and reproduce outside the host cells. Number C, tuberculosis is a viral disease. And number D, viruses are giant compared to bacteria. Which of the following statements is true regarding the virus? Let me know A, B, C or D, which of the following will be the correct answer of problem number 1. I will wait for a minute or two, then I will check the chat box, okay? See, for answering in the class, there is no negative marking. Whatever you think is the correct answer, let me know. Which of the following statements is true regarding virus? Number A, viruses are not living organisms. Number B, viruses grow and reproduce outside the host cells. Number C, tuberculosis is a viral disease. And number D, viruses are giant compared to bacteria. Which of the following statements is true regarding the virus? Let me check the chat box. Let me see if there is any answer for our problem number 1. Okay, I have received number A and number B as the answer. Let me check if which of the following is the correct one, number A or number B. Okay. So the correct answer for problem number 1 will be, it is number A. Okay, viruses are not living organism. This statement is true regarding the virus. So let us first understand what is the difference between virus and bacteria. So in last class we have learned about the bacteria. Today we will be discussing about the virus. Before starting that, let us understand what are the key differences between these two common pathogens. So bacteria, viruses are not living organisms. Okay, this is the very important thing. But bacteria are the living organisms. Why we are saying that the viruses are not living organisms? Viruses only grow and reproduce inside the host cell they infect. When found on outside the living cell, the viruses are dormant. Their life therefore requires the hijacking the biochemical activities of a living cell. Whereas bacteria are living organisms that consist of single cell that can generate energy, make its own food, move and reproduce, typically by binary fission. This allows bacteria to live in many places like soil, water, plants, and the human body and serve many purposes. The viruses are submicroscopic and bacteria are giant compared to the viruses. A viral infection is systematic. Viruses infect the host cell and then multiply by the thousands, leaving the host cell infecting other cells of the body. Whereas the bacterial infection is usually confined to a part of the body described as a localized infection. Infections may be caused by the bacteria or by the toxin or endotoxin produced. Now, viruses, systematic diseases caused by viral infection include influenza, measles, polio, AIDS, and COVID-19. For bacteria, bacterial diseases include the pneumonia, tuberculosis, tetanus, and food poisoning. The general characteristic of the viruses obligate and obligate, intracellular, and unable to self-replicate. Now, once inside the living cell, viruses induce the host cell to synthesize virus particle. The genome is either DNA or RNA, single or double-stranded. Viruses do not have the system to produce ATP. That is why. So this is very important. The viruses do not have any system that can produce ATP, and that is why they are not living organism, because all the living organism has the capability of producing the ATP. The viruses range of the size of 25 to 270 nanometer. Viral tropism. The classification of viruses are based on the nucleic acid type, size and shape of the virion, and the presence and absence of the envelope. The virus structure, one is the virion, the entire viral particle; capsid, the protein code that encloses the genetic material of the virus; capsomere, the protein subunit that makes up the capsid; nucleocapsid, the composed of capsid and genetic material; the envelope is the outer coating composed of the phospholipid bilayer, which is composed of the viral encoded glycoproteins and sometimes viral encoded matrix protein. The envelope is derived from the host cell's membrane. Some viruses use the plasma membrane, whereas others use the endoplasmic reticulum, Golgi, and nuclear membrane. Naked nucleocapsid are the virus with the no envelope. So this is the example. This is the example of the naked nucleoplastic virus, where there is a genetic material or nucleoside, and outside there is a capsid. And this is the involved in or enveloped virus, which is there is a envelope which is protein coating outside the capsid of the virus. Okay, so these are the general characteristics of the virus. Now let us go back to our question, which says which of the statements are true regarding the virus. So number A, viruses are not living organism, which is true. Viruses grow and reproduce outside the host cells. This is not true, because we know that hijacking the host cells system and their various cellular mechanisms are the only way the viruses can grow and reproduce. Okay, tuberculosis is a viral disease. This is also false. Okay, now because tuberculosis is a bacterial disease. In the last class we have learned about the tuberculosis of the bacteria which causes it, and the discussed about it. Then viruses are giant compared to bacteria. This statement also false. Viruses are the submicroscopic, whereas the bacteria are quite giant compared to the viruses. Okay, so these are the differences between the bacteria and virus, and these are the general characteristics of the virus. Okay. Now let me go to the next problem. If you have any doubt or any query after any question, please let me know. Okay. If any question have you feel there is a you do not understand a particular question, please let me know. I will repeat that particular question one more time. Now let me go to the problem number 2 then, if there is no doubt. Problem number 2 says which of the following statements is false regarding the characteristic of the good protein target for antiviral drugs. Your options are: number A, it should be important to the life cycle of the virus; number B, it should be a little resemblance to the human proteins; number C, it should be important in the late stages of the virus life cycle; number D, it should be common to different types of virus. Which of the following statement is false regarding the characteristic of a good protein target for the antiviral drugs? Number A, it should be important to the virus life cycle; number B, it should be a little resemblance to the human protein; number C, it should be important to the late stages of the viral life cycle; and number D, it should be common to different types of virus. Which of the following will be the correct answer? Let me know. A, B, C, or D. Again, let me wait for a minute or two, then I will check the chat box. Okay, meanwhile, let me know A, B, C, or D, which of the following will be the correct answer for our problem number 2. Which of the following statement will be false regarding the characteristic of the good protein target for an antiviral drug? Number A, it should be important to the life cycle of the virus. Number B, it should be a little resembles to the human proteins. Number C, it should be important in the late stages of the virus life cycle. And number D, it should be common to different types of viruses. Let me check the chat box now. Okay, I have received two answers, number C and number B. So again, I have received two different options as the answer, number C and number B. Okay, let me check which of the following will be the correct answer. Second. Okay. So the correct answer will be it is number C. Okay, it should be important in the late stages of the virus life cycle. This statement is false. Other statements are true. Okay, like it should be important to the life cycle of the virus. It should be a little resembles to the human proteins, and it should be common to different types of virus. These statements are true. Now let us understand about the antiviral drugs and what is the key therapeutic features and good. So for good drug targets are the proteins which are likely to have the following characteristic. Okay. So before that, let us understand what is the mechanism that can be done. Used for developing an antiviral drug. So antiviral drugs are specialized medication designed to manage the viral infection by interfering with the virus's ability to replicate and spread. Unlike antibiotics, which often treat multiple types of bacteria, most antivirals are highly specific and only effective against one or few closely related viruses. Okay, so the core mechanism of action, antivirals typically target specific stages of the viral life cycle within the host cell. What is the mechanisms? Inhibition of the entry and attachment, blocking the viruses from binding to or entering the healthy host cell. Inhibition of uncoating, preventing the virus from releasing its genetic material once inside the cell. Okay. Then genome replication blockage, mimicking the building blocks or nucleosides of the DNA or RNA to halt the replication process. For example, acyclovir for herpes, remdesivir for COVID-19. Now, protease inhibition, blocking the enzyme protease that cut the viral protein chains into functional pieces and preventing the assembly of new mature virus, release inhibition, preventing newly formed viruses from leaving the host cell to infect others. For example, neuraminidase inhibitors like Tamiflu. So these are the core mechanisms of action. What are the key therapeutic features? Symptom management, not always a cure. While some infection like flu can be cleared, other like HIV, hepatitis, herpes are chronic. Antivirals make this virus latent or inactive rather than eliminating them entirely. Selective toxicity, they are designed to target viral protein that are different from the human protein to avoid harming the host healthy cells. Time sensitivity, they are most effective when started shortly after symptoms begin. For example, 48 hours for the flu. Resistant potential, viruses can mutate rapidly, leading to the antiviral resistance. This is why drug cocktails and combination therapies are often used for infection like HIV. So the good drug target are the proteins which are likely to have the following characteristic. Number one, they are important. So these are the very important. These 4 points are very important. Okay, good drug targets are the proteins which are likely to have the following characteristic. They are important to the life cycle of the virus such that their inhibition or disruption has a major effect on infection. They bear little resemblance to the human protein, thus increasing the chances of good selectivity and minimal side effect. They are common to a variety of different viruses and have a specific region which are identical to its amino acid combination. This makes the chances of developing drug with a broad antiviral activity more likely, and they are important to the early stages of the viral life cycle, which means their disruption and inhibition reduces the chances of symptoms and of the virus spreading rapidly through the body. So this is important. That is why this Statement number C is false, which says that it should be important for the late stages of the virus life cycle. No, they should be important in the early stages of the viral life cycle, which means their disruption or inhibition will reduce the symptom and viruses spreading through our body. Okay, so number C was the correct answer for problem number 2. Now let me go to the next problem. Problem number 3 says identify the correct order of the viral infection in the host cell. Your options are number A, attachment followed by uncoating, then penetration, then release, then assembly, and then biosynthesis. Number B, attachment followed by penetration, then uncoating, then biosynthesis, then release, and then assembly. Number C, attachment, then biosynthesis, then penetration, then uncoating, then assembly, and then release. And number D, attachment followed by penetration, then uncoating, then biosynthesis, then assembly, and then release. Which of the following is the correct order of viral infection in the host cell? Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 3? You have to identify the correct order of viral infection in the host cell. Your options are number A, first is the attachment, followed by uncoating, followed by penetration, followed by release, followed by assembly, followed by biosynthesis. Number B, attachment, then penetration, uncoating, biosynthesis, released, and assembly. Number C, attachment, biosynthesis, penetration, uncoating, assembly, and release. And number D, attachment, penetration, uncoating, biosynthesis, assembly, and release. Which of the following is the correct order of the viral infection in the host cell? Let me know A, B, C, or D. Which of the following be the correct answer for problem number 3? Which will be the correct order of the viral infection in the host cell? I will wait for a minute, then I will check the chat box. Even if your answer is wrong, it does not matter, but please try to answer the question. Okay, otherwise I won't understand if you are following the class or not. Let me check the chat box. Okay, I have received only a one answer, number C, for the answer for our problem number 3. Okay, now I have to check if number C is the correct answer or not. So the correct answer will be, it is number D. Okay, attachment, then the first step is the attachment, then it will be followed by the penetration, then uncoating, then biosynthesis, then assembly, and then release. Okay, so this is the correct order of the viral infection. So what are the steps of the viral infection? Most viruses follow similar series of steps in the viral reproductive cycle: attachment, penetration, uncoating, replication, assembly, and release. Once inside the host cell, the viral DNA or RNA is released and can begin to interact with the internal cell machinery. Viral DNA can direct transcription. Viral DNA can direct transcription from the viral mRNA, which is then translated into the viral proteins. Viral RNA can be used as template to make more viral mRNA and proteins. RNA retrovirus can use the reverse transcription to make the viral DNA, which is then transcribed and translated. Regardless of the mechanism, the objective is the same: more virions. New virions are released when damage to the host cell causes lysis, and when the host cell undergoes apoptosis or programmed cell death. Some viruses are released through budding, which releases one virion at a time and spurs the host cell. Okay, so this is the correct order: attachment, penetration, uncoating, replication, assembly, and release. So let us understand through the life cycle of a DNA virus, such as the herpes simplex. Okay, so this is our host cell, right? So this is a virion. Okay, so first step is the attachment or the absorption. So first there are some there are some position where this virion can attach to the cell, and then after that penetration will happen through fusion. Okay, as you can see, this is a virion. If it is getting attached, and then it is getting penetrated inside our host cell. After that, what will happen? Now, after that, the uncoating will happen. Okay, this this virus, after it will shed its envelope and it will release the capsid. As you can see here, this nucleocapsid is present here. And after that, what will happen? The biosynthesis, this particular uncoating, after uncoating the viral material, the DNA, mRNA, so all these things will happen, and replication and translation, these things will happen. And after that, what will happen? The assembly, there are the new nucleocapsids which are released, which are synthesis and assembly of the nucleocapsid, and then via budding or lysis, they will be released outside of the host cell. Okay, so these are the particular methods or particular steps by which this viral infection happens. So remember, what are the steps? The steps are the adsorption or attachment, followed by penetration, then uncoating of the genetic material, then biosynthesis of the genetic material or new virions, then assembly of the virions, and then release outside the host cell. Okay, so these are the steps that are followed. Now let me go to the next problem. Problem number 4. It says, which of the following enzyme is responsible for incorporating viral DNA with host DNA? Your options are number A, reverse transcriptase, number B, protease, number C, integrase, and number D, DNA polymerase. Which of the following enzymes is responsible for incorporating viral DNA with host DNA? Reverse transcriptase, protease, integrase, or DNA polymerase? Which of the following enzymes is responsible for incorporating viral DNA with host DNA? Reverse transcriptase, protease, integrase, or DNA polymerase? What will be the correct answer for problem number 4? Let me know. Let me wait for a minute. Then I will check the chat box. Meanwhile, read the question carefully and read the options and then let me know. Okay? Which of the following enzyme is responsible for incorporating viral DNA with host DNA? Reverse transcriptase, protease, integrase, or DNA polymerase? Which of the following enzymes is responsible for incorporating viral DNA with host DNA? Let me check the chat box. Let's see if I find any answer there. Okay, there is no answer for problem number 4. Okay, it is fine. Show you the correct answer and let us discuss it. Okay. So the correct answer will be number C, integrase. Integrase is the enzyme which is responsible for incorporating viral DNA with host DNA. Okay. So this is the life cycle that I have already shown you in our last slide also. Let us discuss it one more time. So first let us understand, so this is a HIV particle or HIV viral particle of HIV. Okay, so this is the structure of the HIV particle. So inside this is the envelope, this is the outer protein. Okay, then we have some cellular, this GP120 or GP41. These actually help for the attachment to the host cell. Then we inside the cell we have this capsid. Okay, and then we have this various enzymes like integrase, reverse transcriptase. Okay, and this is the envelope, this is the capsid which I have already discussed. Okay, so this is the structure. So now how this the life cycle of the HIV in the host cell? The first is the absorption. Okay, so by this receptor, this particular attachment happens of the HIV particle with the host cell. Then penetration, this particular virus particle is taken inside the cell, and after that there are the RNA of the viral RNA, which is this in this after the reverse transcriptase is synthesized the RNA DNA hybrid, and then single strand DNA is synthesized from this, and after that reverse transcriptase, this actually converted to the double strand or normal DNA. And then we have this enzyme. Let me highlight this. This is the enzyme integrase. This enzyme, what does it do? It incorporates the viral DNA with the host DNA. So as you can see here, there the black one is the host DNA and the cyan one is the viral DNA. And integrase, what this is doing, it is incorporating a viral particle or viral DNA to the host DNA. Okay. And then from that viral proteins are synthesized and new viral particle are released via the budding process. So this is the important. So there are various enzymes which are important in the life cycle of the virus. Reverse transcriptase is one of these, and then we have this integrase and other enzymes are also there. So remember, integrase is an enzyme which incorporates this viral DNA to the host DNA. So in biology, integrase is an enzyme that facilitates the integration of the genetic material from one DNA molecule to another. It is more famously known for its role in the HIV life cycle, where it inserts viral DNA into the host cell's genome, but it also plays a critical role in the bacterial evaluation and antibiotic resistance. Viral integrase or retroviruses like HIV. In retroviruses, integrase is essential for replication. It performs two primary chemical reactions to ensure the viral genome gets permanent part of the host cell. 3' processing in the cytoplasm, the enzyme removes 2 or 3 nucleotides from the 3' end of the viral DNA to expose the reactive 3' hydroxyl OH group. Strand transfer, once inside the nucleus, enzyme uses 3' OH groups to cut the host chromosomal DNA and covalently link the viral DNA to the target site. Medical significance, because of humans do not have the equivalent enzyme, integrase is the prime target for the HIV treatment, known as the integrase strand transfer inhibitors, or such as the raltegravir and dolutegravir. So these are the examples of the integrase inhibitor. So whenever we are discussing about any kind of therapeutic agent, for example, for virus, for cancer cell, or for antibacterial, we need to find some kind of target or enzymes which are not present in the host cell. For example, this integrase enzyme, what does it do? It is combining the viral DNA with the host DNA. So in the healthy cell, in the host cell, there is no integrase. Only this enzyme is present inside the virus particle. That is why in designing a compound which can inhibit this enzyme, it will not be harmful for our own system, because in our own system we do not have that particular enzyme, right? So that is why this kind of enzyme or this integrase is a very prime target for the HIV treatment, especially, and this is known as the integrase transfer inhibitor, or the abbreviated from is the INSTI, such as the raltegravir and dolutegravir. So this is an example for the integrase inhibitor. Okay, is it clear? Do you have any doubt or query? Please let me know. Otherwise, I will move to the next problem. Okay. Let me know via chat box if you have any doubt or query. I will repeat that part, whatever you feel is the problematic. Okay. So let us go to the next problem. If there is no issue with this question, let me first check the chat box. Any doubt or query? Okay. Now let me show you the next question. Let us identify the, or let us understand the next problem. So the next problem number 5 says, which of the following is the correct set of enzymes for the transformations mentioned below? Okay. Number A, thymidine kinase followed by esterase. Okay, sorry. Okay, sorry. I think I did a pretty mistake. Okay, I forgot to give you the transformation here. If I do, okay. Fine. The question was actually the this transformation of this to this, actually. Okay, the conversion of famciclovir to penciclovir. This transformation, the question was related to this. The correct set of enzymes you have to identify for this to this. Okay, so I forgot to give you the particular thing. Okay, but let it be. So this is actually a antiviral drug, famciclovir. Okay, and this is converted to this hydroxyl part. This is the by the esterase, where the esters are cleaved and we get our alcohol or OH groups here. Okay, then aldehyde oxidase is present. Aldehyde oxidase is used. What does this aldehyde oxidase do? Actually, basically, this is oxidizes in this position. Okay, and then viral thymidine kinase comes and it attached to one phosphorylate this one, and then cell kinases are triphosphorylate this one. So I just the question that I intended to give was the conversion of this to this. So this was the question. Okay, it's fine. Uh, I think it is pretty understandable. You had to identify which enzymes are involved. So basically, this to this, as you can see clearly, one ester is converted to the alcohol, right? So you have to identify the correct ester. Was the correct set of enzyme is the esterase. So first is this the esterase, followed by the cell kinase, and then esterase followed by the aldehyde kinase. These two options has to be correct. And among these two, then you will see the conversion is like this. So you will see there is a oxidation type of thing happens. So the correct enzyme will be aldehyde oxidase because the oxidation is happening, right? Let me go to the next problem. Problem number 6 says which of the on which receptor of T cell surface the HIV binds. Your options are number A CD4, number B CD6, number C CD7, and number D CD2. On which receptor of T cell surface the HIV binds? On which receptor of T cell surface HIV binds? CD4, CD6, CD7, or CD2? Let me know A, B, C, or D which of the following will be the correct answer of problem number 6. On which receptor of T cell surface the HIV binds? CD4, CD6, CD7, or CD2? I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D which of the following you think is the correct answer. If you have paid attention to our fourth question, there was the receptor was mentioned actually in that particular life cycle of the HIV particle that I have discussed that time. You have to identify correct receptor of the T cell surface where the HIV binds. CD4, CD6, CD7, or CD2. Let me check the chat box. Let's see if you have any answer for the problem number 6. Okay, no answer. See, even if your answer is wrong, it does not matter. So please try to answer the questions, okay? Even if your answer is wrong, it's fine. Okay, let us understand what is the correct answer and let's discuss it. The correct answer will be number A, CD4, okay? So this is the particular how the HIV infection happens. So first is the attachment of the binding phase. So as you can see, so the C4 is the cellular receptor. Let me just highlight it. C4 is the receptor by which CD4 is the receptor by which this particular HIV particle binds to the surface of the host cell. So this is the HIV virus. Okay, as you can see, we have the important enzymes inside the capsid. We have the integrase RNA, we have RNA, we have integrase, we have reverse transcriptase and protease enzymes. And there outside that we have the capsid, which is protecting all the viral genetic materials. And after that we have this envelope which is circulating the capsid. Okay, and on this we have this GP41 and GP120. They basically identify our CD4 receptor and binds to the receptors. Okay, and after so the binding on the surface of the T cell HIV binds to a CD4 receptor and one or two co-receptors like CXCR4 and CC or CCR5. So we have CD4 as a receptor and also we have one of the two co-receptors CX CR4 or CCR5. So two co-receptors are also there, CXCR4 and CCR5. One of the two co-receptors and CD4, they will be engaged in binding with our HIV particle. And then fusion will happen. So as I have already mentioned it couple of times, the first penetration, then we have this penetration or fusion. And when it is fused, as you can see, the capsid is goes inside and it releases all the genetic material. We have the RNA, we have the DNA from the with the help of the reverse transcriptase, and then we have this viral RNA. So reverse transcription, what happens? The whole single stranded HIV RNA is converted into the double stranded HIV DNA by the reverse transcriptase enzyme, then followed by the integrase, which cuts the cell DNA and inserts the viral DNA inside the healthy cell DNA. As you can see, this is the host DNA, and integrase is cutting and inserting a particular viral DNA inside it. And then long RNA chains of the integration. After HIV DNA enters the cell's nucleus, the enzyme integrase cuts the cell's DNA and inserts the HIV DNA into it. Then is the transcription and translation. The enzyme RNA polymerase makes RNA copies of the DNA. HIV RNA is either inserted into the new virus particle or processed and translated into the HIV proteins. Then assembly, long protein chains are cut into the individual proteins by the enzyme HIV protease, and new virus is assembled with these proteins and HIV RNA. And then new viral particle are synthesized and released. A new viral particle is released from the host cell, taking it into the part of the cell's membrane and capable of the infecting the other cell. Okay, so these are the all the steps. So you have to remember that the receptor is the CD4 and co-receptors are CXCR4 and CCR5, which are involved in the the attachment of the HIV particle to the host cell. So the role in what is the role exactly? What is the role of this? If you are had to write, so what is the role of the CD4 receptor in the HIV? Infection. HIV hijacks the CD4 receptors to infect the and untimely destroy the T helper cell, leading to the immune deficiency that characterized in AIDS. Binding the HIV envelope protein GP120 binds directly with high affinity to the CD4 receptors on the T helper cell surface. Co-receptor engagement. This binding causes the conformational change in the viral protein, allowing it to bind to the second co-receptor on the host cell, either CCR5 or CXCR5 for fusion and entry. The dual receptor binding facilitates the fusion of the viral envelope with the host cell membrane, allowing the virus to enter the cell. Immune depletion. As HIV replicates and kills CD4 plus T cell, the body's immune system is progressively weakened and making it susceptible to the opportunistic infection. Monitoring the CD4 count is standard part of HIV treatment to assess the health of the patient's immune system. Okay. So this is the role of the CD4 receptor in the HIV infection. Now let me go to the next problem. Problem number 7 says, which of the following is not true? Your options are: number A, DNA virus has lower mutation rate. Number B, reovirus or RNA virus. Number C, RNA virus mutation occurs inside the nucleus. And number D, double-stranded DNA viruses are most common. Which of the following is not true? Number A, DNA virus has lower mutation rate. Number B, reovirus or RNA virus. Number C, RNA virus mutation occurs inside the nucleus. And number D, double-stranded DNA virus are most common. Which of the following statement is not true? Whenever there is a true-false kind of question, I always say read options carefully and then let me know A, B, C, or D, which of the following will be answered. Because sometimes this true-false, correct, incorrect kind of questions can be a little tricky. So read all the options carefully and then let me know A, B, C, or D, which of the following will be the correct answer for problem number 7. Number A, DNA virus has low mutation rate. Number B, reovirus or RNA virus. Number C, RNA virus mutation occurs inside the nucleus. Number D, double-stranded DNA viruses are most common. Which of the following statement is not true? Which of the following is not true? DNA virus has lower mutation rate. Number A, number B, reovirus or RNA virus. Number C, RNA virus mutation occurs inside the nucleus. Number D, double-stranded DNA virus are most common. Let me check the chat box. Okay, still no answer. See, even if your answer is wrong, it's fine, but try to answer the questions. Okay, otherwise I won't understand if you are following the class or not. Okay, let me show you what is the correct answer. So the correct answer will be number C, the statement which is not true. RNA virus mutation occurs inside the nucleus. This statement is not true. So let us understand first and let us see what are the differences between the RNA and DNA virus. So the DNA viruses are the viruses that possess DNA as the genetic material, whereas the RNA viruses are the viruses that possesses RNA as the genetic material. The common type, the most common type is the double-stranded DNA virus, and the most common RNA type virus are the single-stranded RNA virus. The genome size of the DNA virus are larger, and for RNA virus it is the smaller. The replication occurs inside the nucleus for the DNA virus. The replication occurs inside the nucleus, whereas in the case of RNA virus, the replication occurs in the cytoplasm. So number C, which says the RNA virus mutation. Okay, this occurs inside the nucleus, so it is not. So the mutation rate, lower mutation rate for the DNA virus, and for RNA, it is a higher mutation rate. Accuracy of replication, slow and accurate replication in case of the RNA one, but the error-prone replication happens in the RNA one, but the accurate replication happens in the DNA virus. Types, double-stranded DNA virus and single-stranded DNA virus, and the examples, and the examples of RNA virus, double-stranded RNA virus, positive sense single-stranded RNA virus, negative strands single-stranded RNA viruses are the types. Examples, adenovirus, herpes virus, pox virus, parvovirus, and hepat DNA virus are like the example of DNA virus. Reovirus, piconavirus, togavirus, these are examples of the RNA virus. So basically, these are the examples. If it is not possible to remember all of this, just remember one or two, because if the question asks that give an example of the DNA or RNA virus, you should be able to answer that. So example, pox virus, herpes virus, adenovirus, HepaDNA virus, and papilloma virus, papilloma virus are the DNA virus, whereas the myxovirus, paramyxovirus, rubella virus, rhabdovirus, these all are examples of the RNA viruses. Okay, so these are the examples, and these are the key differences between the DNA and RNA virus. Now let me go to the next problem. Problem number 8 says, which of the following statements is false regarding interferons? Your options are number A, they are classified as broad spectrum antiviral agent. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. And number D, immunomodulators are used to induce the interferon production. Which of the following statements is false regarding interferons? Again, I will repeat. Number A, they are classified as a broad spectrum antiviral agent. Number B, they are small natural proteins. Number C, beta interferons are generated from lymphocytes. Number D, immunomodulators are used to induce interferon production. Which of the following statement is false regarding interferons? I will wait for a minute, then I will check the chat box. Let me know. Read the options carefully and then answer. Which of the following statement is false regarding interferons? Number A, they are classified as a broad spectrum antiviral agent. Number B, they are small natural protein. Number C, beta interferons are generated from lymphocyte. And number D, immunomodulators are used to induce interferon production. Which of the following statement is false regarding interferon? Let me check the chat box. Let's see if there is any answer. Okay, I have received one answer saying that number D is the correct answer. Okay, let me check if number D is the statement which is false regarding interferons, or is there any other answer. So the correct answer will be number C. Okay, beta interferons are generated from lymphocyte. This statement is false. Okay, it is fine. At least I have received an answer after a very long time. So good. So basically, interferons are small natural proteins that were discovered in 1957, which are produced by the host cell as response to the foreign invaders. Once produced, interferons inhibit the protein synthesis and other aspects of the viral replication in the infected cells. In the other words, they shut the cell down. This can be described as an intracellular immune response. Administering interferons to patients have seen as possible approach for treating the flu, hepatitis, herpes, and colds. There are several interferons which are named according to their source. Alpha interferon from the lymphocyte. So these things are important. Okay, the names of the or the nomenclature of the interferons, because okay, so alpha interferons, they generated from the lymphocyte, beta interferons from the fibroblast, and gamma interferons from the T cell. Alpha interferons are also known as the alpharon or IFN alpha, is the most widely used for the used of the three types. In the past, it was difficult and expensive to isolate interferon from the natural cell, but recombinant DNA techniques allow this production to be genetically engineered interferons in larger quantities. Recombinant alpha interferon is produced in three main forms. The alpha 2A or 2B are the normal form, and alpha con 1 is the unnatural form. They have proved successful therapeutically and can be, but they can have serious side effects. At present, alpha interferon is used clinically against hepatitis B infection. Okay. This is also important. It is clinically used against the hepatitis B infection. It is also used with the ribavirin against the hepatitis C infection. Interferon production in the body can also induce the agents named as immunomodulator. One such example is the Avridine, okay, which is used as a vaccine adjuvant for the treatment of the animal disease such as for the foot and mouth. Imiquimod also induces the production of alpha interferon as well as other cytokine to stimulate immune system. It effective against the genital warts. So antiviral actions of the interferon. So basically, first is the viral DNA. It is of the infected cell. It goes to the healthy cell. Okay, and after the penetration, what happened? It releases its viral genetic material, the viral DNA, RNA, and this is the interferon mRNA. So first is the viral RNA from the infecting cell inside the enters the cell, and the infecting virus replicates into the new viruses. This infecting virus can induce the host cell to produce the mRNA or IF interferon mRNA, which is translated into the alpha and beta interferon. So once the alpha and beta interferon are released from the virus infected, so this cell is virus infected. Once it is virus infected, this induces the host cell to produce. This is the interferon. This is the alpha and beta interferon, and the interferon is released by the virus infected host cell to plasma membrane or the nuclear membrane receptors of the uninfected neighboring cell and induce them for synthesize the antiviral proteins or AVPs. This induces the oligoadenylate synthase and protein kinase. New viruses are released, and these viruses, once they go to the neighboring host cell, this antiviral protein degrade the mRNA of the viral mRNA and inhibit the protein synthesis, thus interfere with the viral replication. So this is the mechanism of action of the interferons. So first, when a cell is getting infected, interferons are generated from the virus infected cell, which goes to the neighboring cell to alert the neighboring cell, and the neighboring cell it produces antiviral proteins in presence of the interferon, and this antiviral proteins kills the viral particle, means inhibits the viral mRNA and protein synthesis. That's how they inhibit this virus. Okay, that is why the antiviral. That is how the antiviral action of the interferon happens. Okay. Now let us go to the next problem. Problem number 9 says, which of the following statement is false regarding adamantins? Your options are: number A, they are known as ion channel disruptors. Number B, they inhibit the replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change in the concentration, their mechanism of action changes. And number D, the virus can't mutate in the presence of the adamantine. Which of the following statement is false regarding adamantine? Number A, they are known as the ion channel disruptor. Number B, they inhibit the replication of the influenza C virus by blocking the matrix protein channel. Number C, with the change in the concentration, their mechanism of action changes. Number D, the virus cannot mutate in the presence of the amantidine. Which of the statements or following statements is false? Read options carefully and then let me know A, B, C, or D. Which of the following is this thing is the false statement regarding the adamant tense? I will wait for a minute, then I will check the chat box. Okay. Meanwhile, let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following statements is false regarding adamantines? Number A, they are known as ion channel disruptor. Number B, they inhibit the replication of the influenza A virus by blocking the matrix protein channel. Number C, with the change in concentration, their mechanism of action changes. And number D, the virus cannot mutate in presence of the amantidine. Let me check the chat box. Let's see if there is any answer for problem number 9. Okay, there is no answer for problem number 9. Okay, it's fine. Let me show you the correct answer and then let us understand what is the role of the adamantins in the antiviral drug discovery. So basically, the correct answer, number D, which is false, which says that the virus cannot mutate in presence of amantadine. This is false. So basically, this is the structure of the adamantines. This is the amantadine and this is the rimantadine. Okay. So the adamantines were discovered by random screening and are most earliest antiviral drugs used in clinically against the flu, decreasing the incidence of the disease by 50 to 70%. Amantadine and rimantadine are related adamantine with the similar mechanisms of action and can inhibit the viral infection in two ways. At low concentration, less than 1 microgram per ml, they inhibit the replication of the influenza A viruses by blocking a viral ion channel protein called matrix M2 protein. At higher concentration, the basic nature of the compounds became important and they buffer the pH of the endosomes to prevent the acidic environment needed for the HA to fuse with the viral membrane with the endosome. These mechanisms inhibit the penetration and uncoating of the virus. Unfortunately, the virus can mutate in presence of amantadine to form resistant variants. Amantadine binds to the specific regions of the M2 ion channel, and resistant variants have mutations which alter the width of the channel. Research carried out find analogs which might still bind to this mutant proved unsuccessful. Work has also been carried out in an attempt to find analog which might affect the ion channel and pH levels at a comparable concentration. This is focused on secondary and tertiary amines with increased basicity as well as alteration of the structure to reduce the activity for the ion channel. The rationale is that the resistant flu variants are less likely to be produced if the drug acts on the two different targets at the same time. Rimantadine was approved in 1993 as a less toxic alternative to amantadine for treatment of influenza A. Unfortunately, neither agent is effective against the influenza B as the virus does not contain a matrix M2 protein. Side effects are also a problem, possibly owing to the effects of the host cell ion channels. Okay. So these are the all about the adamantane, which are the ion channel disruptor. Two are the most common, which are have similar methods of action. One is amantadine and one is rimantadine. But it is not a very good drug target because the virus can mutate in presence of these, and also they have some toxic side effect. And also they are not active against the influenza virus because it does not have the matrix M2 protein. Okay, so these are the all about the amantadines element. So now let us go to the next problem. Problem number 10 says the only DNA antisense molecule that has been approved as antiviral drug is, your options are number A, nevarapine, number B, formivirsen, number C, darunavir, and number D, zerovodine. The only DNA antisense molecule that has been approved as antiviral drug is number A, nevirapine, number B, formivircin, number C, darunavir, and number D, zidovudine. Which of the following will be the correct answer of problem number 10? Let me know. Maybe C or D. The only DNA antisense molecule that has been approved as antiviral drug is nevirapine, formivircin, darunavir, or zidovudine. What will be the correct answer for problem number 10? Which of the following is the only DNA antisense molecule that has been approved as antiviral drug? Let me wait for a minute and then I will check the chat box. Please let me know whatever you think is the correct answer. Even if your answer is wrong, it is fine. But try to answer the question. The only DNA antisense molecule that has been approved as antiviral drug is, let me check if there is any answer in the chat box. Okay, I have received one answer saying number A is the correct one. Let me see if number A is the correct one or not. So the correct answer is fact number B. Formivircin is the only DNA antisense molecule that has been approved as antiviral drug. So the antisense therapy, so this is the structure of the formivircin, okay. So is the first and so far the only DNA antisense molecule that has been approved as an antiviral drug. It consists of 21 nucleotides with the phosphonothioate backbone rather than a phosphate backbone to increase the metabolic stability of the molecule. The drug blocks the translation of the viral RNA and used against the retinal inflammation caused by CMV in the AIDS patient. Because of the high polarity, it is administered as an ocular injection or intravitreal injection. Okay, because it has a very high polarity. Because as you can see there, it consists of 21 nucleotides. Okay, so this is the antisense drug, the oligonucleotide, which degrades and the no translation can happen. So formivircin is marketed as the Vitravin, holds a unique place in medicinal history as the first ever antisense oligonucleotide drug to receive FDA approval in 1998. It was designed to treat the cytomegalovirus retinitis, a serious eye infection that can lead to blindness, particularly immunocompromised patients with AIDS. Mechanism of action: formivircin works through an gene splicing mechanism rather than interacting with the proteins like the traditional drugs. Antisense binding: it is a 21-mer phosphorothioate oligonucleotide with a sequence complementary to the mRNA of the CMV E2 immediate early 2 gene. Protein inhibition: by binding to this specific mRNA, it is physically blocks the translation process, preventing the virus from producing the essential IE2 protein required for the viral replication. Sequence independent effects: interestingly, studies suggest that it may also have the non-specific effects such as the preventing the virus from attaching to the host cell. Why it was discontinued? The formivircin is often cited as a drug that was too successful for its own market. The rise of the HAART, the introduction of the highly active antiretroviral therapy for the HIV and AIDS dramatically improved the patient's immune system. Market disappearance as immune response recovered. Cases of CMV retinitis plummeted. With fewer patients needing the drug, it became commercially unviable and withdrawn from the market from like in 2002 and 2006. Despite its withdrawal, the success of the Vitravin proved that antisense technology could work in humans, paving the way for the modern RNA-based therapies like patisiran. Okay. So this is all about the formivirsin, and a very this is very important because it is the it is the so far is the first ever antisense molecule that to be reached in the market for the treatment of a particular antiviral infection. Okay. So now I will go to the next problem. Problem number 11 says identify the statement that is incorrect. Your options are number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number B, cancer can result when one or more cell checkpoints are malfunction. Number D, G2 phase occurs between the S and M phase. Which of the following statement is incorrect? Number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, a DNA synthesis checkpoint is present. Number C, cancer can result when one or more of the cell cycle checkpoints malfunction. Number D, G2 phase occurs in between the S and M phase. Which of the following statement is incorrect? Again, I will wait for a minute or two, then I will check the chat box. Let me know A, B, C, or D, which of the following statement is incorrect. Which of the following statement is incorrect? Number A, DNA synthesis checkpoint exists in the growth phase of the cell cycle. Number B, in the synthesis phase, the DNA synthesis checkpoint is present. Number C, cancer can result when one or more cell cycle checkpoints malfunction. Number D, G2 phase occurs between the S and M phase. Which of the following statements is incorrect? Let me check the chat box. Let's see if there is any answer for our problem number 11. Okay. I have received one answer saying number B is the correct one. Let me check if number B is the statement which is incorrect. Okay, so the correct answer is number A, means the statement which is incorrect. Okay, so DNA synthesis checkpoint exists between in the growth phase of the cell cycle. This is false. Number B actually the correct statement. Okay, in the synthesis phase, the DNA synthesis checkpoint is present. Now let us understand this is the cell cycle and this is the checkpoint. So in our cell cycle we have several phases. Majorly we have four different phases: G1, S, G2 and M. G1 is the growth phase, S is the synthesis phase, G2 is the growth phase 2 and M is the mitosis phase. Okay, so basically in the in each and so three checkpoints are present. So the following diagram references the various checkpoints. So three different checkpoints are present in the cell cycle. First is the cell growth checkpoint. This occurs at the end of the growth phase 1 or G1. Here it occurs, okay, at the end of the growth phase 1. It may checks whether the cell is big enough and has made proper proteins for the synthesis phase. If not, the cell goes through a resting period or G0 until it recovers and it is ready to divide. Then there is a DNA synthesis phase, and here we have the DNA synthesis checkpoint which occurs inside this synthesis phase. It checks whether the DNA has replicated correctly. If so, the cell continues to the mitosis. Then we have this G2 phase, which is the growth phase 2, and then mitosis phase, And here we have mitosis checkpoint, which occurs in this mitosis phase and checks whether mitosis is complete. If so, the cell divides and the cell cycle repeats. So these are the different phases of the cell cycle and the checkpoints which are present in the cell cycle. So remember, now we have four phases and three different checkpoints. Let me highlight it. One is the cell growth checkpoint. Then we have the DNA synthesis checkpoint. And then we have the mitosis checkpoint. So three checkpoints and four different phases, G1, S, G2, and M. Okay. And the diagram is very important. This cell diagram is very important that you have to remember. Now, so basically, the stalled DNA replication fork will activate the S phase checkpoint where it is ensured a proper replication of the genetic material. If this checkpoint is somehow missed, another checkpoint at G2 will reassure proper replication and devoid of any DNA damage prior to initiating the mitosis cycle. So this is the cell cycle. One is the G1 phase, then GS phase and G2 phase. So the cancer result basically from one or more of this check cycle check points malfunction. Okay. So remember, so what does these checkpoints check? First, they check for damaged DNA, then unreplicated or damaged DNA, and here it checks for the chromosome misalignment. Okay, so these are the checkpoints and these are the roles of the different checkpoints. Now let me go to the next problem. Problem number 12 says which cyclin is involved in balancing cell growth and division? Your options are number A cyclin A, number B cyclin B, number C cyclin D, and number D cyclin E. Which cyclin is involved in balancing the cell growth and division? Cyclin A, B, D, or E? Let me know the correct answer A, B, C or D, which of which cyclin is involved in balancing the cell growth and division. Again, I will check the chat box after a minute. Meanwhile, let me know A, B, C or D, which of the following will be the correct answer for problem number 12. Which cyclin is involved in balancing cell growth and division? Cyclin A, B, D, and E. Which will be the correct answer? Let me check the chat box. Let's see if there is any answer for problem number 12. Okay, I have received number C as the answer. Let me check if number C is the correct answer or not. So the correct answer is very good. It is number C. Cyclin D is the cyclin that is involved in balancing the cell growth and division. So in the cell cycle, we have different cyclines which are present. In our G1 phase, we have the cyclin D1, 2, and 3, and CDK 4 and 6 is the cyclin dependent kinase. Okay, and cyclin E is present at the end of this particular G1 phase, and we have the CDK2 which controls it. Then we have the cyclin A in the S phase and CDK2. CDK2 and cyclin A are also present in their G2 phase, and CDK1 and cyclin B is present in the M phase. Okay, so cyclin D, it is involved in the cell growth and division, and this is present in the G1 phase. So basically, control of the cell cycle involves a variety of proteins known as the cyclins, okay, and the enzymes known as the cyclin dependent kinase. So cyclins are the enzymes and the cyclins are protein, and the cyclin dependent kinases are the enzymes which are involved in the cell cycle. There are at least 15 types of cyclin and 9 types of CDK, and each has a role to play in the different stages of cell cycle. Example is shown in this figure. This figure is very important where the control of the cell cycle by cyclin and cyclin dependent kinases are shown. Okay, So binding a cyclin to its associated kinase activates the enzyme and serves to move the cell from one phase of cell cycle to another. For example, when the cell is in G1 phase, the decision has to be made whether to move the cell to the S phase and start the copying DNA. This decision is taken depending on the balance of the stimulatory versus the inhibitory signal being received through the signal transduction. If the balance is toward the cell growth and division, there is an increase in the cyclin D. This binds to the cyclin CDK4 and 6 and resulting the complex phosphorylate powerful growth inhibitory molecule known as PRB, which normally binds and inactivates the transcription factor. Phosphorylation alters PRB such that it can no longer bind to the transcription factor, and later the latter is free to bind specific regions of DNA. This results in the transcription of the specific genes, which leads to the production of the proteins capable of moving toward the cell toward the S phase. There is the cyclin E and thymidine kinase. Once cyclin E is produced, it combines with the CDK2 and the complex is responsible for progression from G1 phase to S phase. Other activated cyclin CDK4 complexes are important in the different phases of the cell cycle. For example, cyclin A CDK2 complex is required for progression to the S phase and cyclin B CD1 complex is necessary for mitosis. Okay. So these are the complexes which are necessary. So cyclin D and CDK4 and 6 are required for the growth and then movement of the G1 to S phase. Then the progression through S phase requires cyclin A and CDK2 complex, and cyclin B and CDK1 complex is necessary for the mitosis. Okay. So this is about the cyclins and their importance in the cell cycle. Now let me go to the next problem. Problem number 13 says, what is the general term used to indicate the growth of the new blood vessels? Your options are number A, biosynthesis, number B, apoptosis, number C, angiogenesis, and number D, metastasis. What is the following, which is the general term that is used to indicate the growth of the new blood vessel? Biosynthesis, apoptosis, angiogenesis, or metastasis? Let me know A, B, C, or D, which of the following will be the correct answer of problem number 13. I will check after 2 minutes. Okay, let me know. What is the general term used to indicate the growth of the new blood vessels? Biosynthesis, apoptosis, angiogenesis, or metastasis? What is the general term used to indicate the growth of the new blood vessels? Biosynthesis, apoptosis, angiogenesis, or metastasis? Let me check the chat box. Let's see if there is any answer for problem number 13 in the chat box. Okay, number C. Let me check if number C is the correct answer or not. So the correct answer will be very good. Number C, angiogenesis is the term that is used to indicate the growth of the new blood vessel. So as a tumor grows, its cancerous cells require a steady supply of amino acid nucleic acid, bases, carbohydrates, oxygen, and growth factor if they are to continue multiply. This means that the tumor has to have a good blood supply as the tumor grows in size. However, it cells becomes more increasingly remote from the blood supply and becomes starved in this of these resources. Oxygen levels also fall, resulting in a state of hypoxia. This is particularly true for the cells in the center of the tumor. As a result of this hypoxia, inducible factors or HIF1 start to build up within the tumor cell, and these factors serves to upregulate genes that promote the survival of the oxygen-starved environment. For example, growth factors are released of the cell such as the vascular endothelial growth factor or VEGF and fibroblast growth factor FGF2, which interact with the receptors on the endothelial cells of the neighboring blood vessels and stimulate these cells to divide and leading to the branching and extension of the existing capillary, a process known as the angiogenesis. So this is the tumor cell, and this is the epithelial cell. So this angiogenesis happens when the tumor cells. This is the new growth of the new blood vessels. Okay, so the angiogenesis is defined as the formation of new capillaries from the pre-existing vascular by the migration and proliferation. And this is the example of the figure how it happens. Okay, so the growth of the new blood vessels, which is known as the angiogenesis. Now let me go to the next problem. Problem number 14 says which of the following statements is untrue regarding the mitomycin? Your options are number A, it is an excellent choice of drug for the solid tumors. Number B, it is activated by biooxidative process. Number C, it is a prodrug. And number D, its mechanism of action, there is a Michael reaction. Which of the following statement is untrue regarding the mitomycin? Number A, it is an excellent choice of drug for solid tumor. Number B, it is activated by biooxidative process. Number C, it is a prodrug. And number D, its mechanism of action, there is a Michael reaction. Let me know A, B, C or D, which of the following will be the correct answer. Which of the following statements is untrue regarding mitomycin? Number A, it is an excellent choice of drug for solid tumors. Number B, it is activated by biooxidative process. Number C, it is a prodrug. And number D, in its mechanism of action, there is a Michael reaction. A, B, C or D, which of the following statement is untrue? I will wait for a minute, then I will check the chat box, okay? Let me know A, B, C or D, which of the following statement is untrue regarding mitomycin? Let me check the chat box. Let's see. Okay, number B is the answer that I have received. Okay, let me check if number B is the correct answer or not. Let me check the chat box. Okay, okay, sorry. Let me check what is the correct answer. Number B is the option that I have received as the answer. Let me check if number B, yes, it is correct. It is activated by biooxidative process. This statement is untrue. Okay, this statement is not correct. So this is the mitomycin, the first step. is the reduction. So as you can see, the first step is the reduction. This is the quinone, and the this conversion of this ketones to the alcohol is the first step. This is the reduction step, right? And then followed by this I mean this movement of the double bond to the this ring closer, and after that we have this ring opening, and this then alkylating agent. It can act as alkylate. This after this ring opening, it is an alkylating agent. So then DNA, it has a NH2 which attacks on this position, and this kind of adductase form. Okay, so basically the mitomycin was discovered in 1950s, and again it is isolated from a class of microorganism known as the streptomyces. It is one of the most toxic anti-cancer drug in the clinical use, and it is another example of prodrug. Of course, we will look into the prodrugs. So we have already I mean we also already have discussed about the prodrugs. So the enzymes can carry out this known as the bioreductive process. So this is a bioreductive process, not a biooxidative process. There are many enzymes inside our body which can do oxidation as well as reduction, and they are known as the oxidoreductase. So when mitomycin encounters a bioreductive enzyme, this quinone here undergoes the reduction to form a diol, and now quinone is actually electron withdrawing group, which we will imagine as a carbonyl group as an electron withdrawing, whereas once we forming diol, it converts to a electron rich, which can promote the rearrangement, and eventually eventually pick up a proton to produce species which can become a substrate for the attack by DNA. As a reduction step involved in the mechanism, it has been proposed that the drug should be more effective against the tumor in an oxidant starved or hypoxic environment such as the center of the solid tumor. Okay, because it is the drug is gets activated by the bioreductive process, so it is very good for the solid tumors inside the tumors, because there is a oxygen deprived or hypoxic environment. That is why it is very good for that kind of tumor. Okay, and the mechanism of action is it is a alkylating agents. Okay, and regarding alkylating agent, I think I have already discussed in our when I was discussing about the nucleic acid as a drug target, so you can go through that material. Now I will go to the next problem. Problem number 15 says, which of the following is not an anthracycline anti-cancer drug? Your options are number A, doxorubicin, number B, epirubicin, number C, darunavir, and number D, all of the above. Which of the following is not an anthracycline anti-cancer drug? Number A, doxorubicin, number B, epirubicin, number A, darunavir, and number D, all of the above. Let me know. Which of the following is not an anthracycline anti-cancer drug? A, B, C, or D. Which of the following will be the correct answer? Let me know. I will check the answers after a minute. So, whatever you think is the correct answer, you can write in the chat box A, B, C, or D. Which of the following is not an anthracycline anti-cancer drug? Doxorubicin, epirubicin, darunavir, or all of the above? Let me check the chat box. Let's see if there is any answer for problem number 15. Okay, number D is the answer that I have received. Okay, let me check if number D is the correct answer for our problem number 15. So the correct answer is actually number C, darunavir. Darunavir is not an anthracycline anti-cancer drug. Okay, so these are the anthracyclines. This is the common structure of the anthracycline. You can see three ring or four rings are together. Okay, and this is the main part, this planar ring. So this part are planar. Okay, so this blue in the bluish part, these are the planar. So these three ring conjugate together is, but this portion is not planar. So regard this, there are several group R1 here, R2 here, R3 and R4. Different groups are present in this as R1, R2, R3 and R4. So these are the these are the examples of the anthracycline: doxorubicin, okay, epirubicin, daunorubicin, and idarubicin. So all these are the anthracyclines. Okay, the rubicin, the remember the rubicin, wherever there is a rubicin, it is an anthracycline drug. But darunavir, it is actually a antiviral agent. Okay, so these are known as the intercalating agents. So I think I have discussed about intercalating agent in our fifth or sixth class. I do not remember exactly, but when I was discussing about the nucleic acid and drug target, there I have discussed about the intercalating drugs. So intercalating drugs contain a planar aromatic or heteroaromatic ring system which can slip into the double helix of the DNA and distort the structure. Once bound, the drug can inhibit the enzymes involved in the replication and transcription processes. Examples include the dactinomycin and doxorubicin. Doxorubicin, previously called as the adriamycin, belongs to a group of the naturally occurring antibiotics called as the anthracyclines and was isolated from the streptomycin butonius in 1967. It is very similar to the structure of the daunorubicin, differing only in one hydroxyl group. However, that has an important effect on the activity of the doxorubicin as one of the most effective anti-cancer agent ever diagnosed. The drug intercalates into the DNA as an example of the topoisomerase 2 poison as it stabilizes the complex formed between the DNA and topoisomerase 2, an enzyme that is crucial for the replication process. It is thought that an excessive number of the stabilized DNA enzyme complexes triggers the apoptosis. Because of these enzymes are active during the cell growth and division, the topoisomerase 2 poisons are most effective rapidly proliferating cells. The second mechanism of action by which the doxorubicin can prove harmful to DNA involves the hydroxyquinone moiety which can chelate iron to form the doxorubicin DNA iron complex. Reactive oxygen species are then generated leading to the single strand breaks in the DNA chain. This mechanism is considered less important than the intercalation with the topoisomerase 2, but it has been implicated in the cytotoxicity of the doxorubicin. A third proposed mechanism involves intercalated doxorubicin inhibiting the helicases, which unveil the DNA into the single strand. A variety of other anthracyclines are used in cancer chemotherapy, mainly daunorubicin, or called as the cerorubicin, daunomycin, and rubidomycin. The second generation anthracycline, epirubicin and idarubicin, are also there. Idarubicin lacks in the methoxy group R1, so it is more polar and its altered metabolism also prolongs the half-life. A third generation of anthracycline is being studied where the ammonium ion, now the sugar ring, is replaced in the azido group and triazole ring. Such compounds that do not appear to be susceptible to the efflux mechanism and leads to the drug resistance and also decreased general toxicity. So this is all about the intercalating agents and the anthracycline class of anti-cancer drugs. Now, the problem number 16 says, which of the following is not a third generation aromatase inhibitor? Your options are number A, anastrozole, number B, letrozole, number C, vorozole, and number D, fedrozole. Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, vorozole, or fedrozole? Let me know A, B, C, or D. Which of the following will be the correct answer? After a minute, I will check the chat box. Okay. Which of the following is not a third generation aromatase inhibitor? Anastrozole, letrozole, vorozole, or fedrozole? Let me check the chat box. Let me see if there is any answer. Okay, there is no answer till now. Okay, let me check which of the following will be the correct answer, and then let us discuss. So the correct answer is number D. Fedrozole is not an third generation aromatase inhibitor. Okay, so basically aromatase inhibitors. Yeah. So aromatase inhibitors tend to be used as the second line drugs for the treatment of estrogen dependent breast cancer. A membrane bound enzyme consists of the two proteins. One is the cytochrome P450 containing heme, and other is the reductase enzyme using NADPH as the cofactor. Aromatase catalyzes the late stage of the biosynthesis of the estrogen from androgen where the aromatic ring is formed. The cytochrome enzyme contains the heme which serves to bind the steroid substrate and oxygen that catalyze the oxidation. Since the enzyme catalyzes the last step of the synthesis, it has been seen as an important target of the design of the anti-estrogen drug. Two types of inhibitors are used clinically: reversible and competitive inhibitors, and irreversible inhibitors or the suicide inhibitors. Aminoglutathione are the early example of reversible competitive inhibitor, but it has disadvantage. It binds to the various cytochrome P450 enzymes and inhibit a range of steroid hydroxylations. This results in undesirable side effects. Drug design based on the aminoglutamate as a lead compound resulted in the more selective inhibitors such as the anastrozole, letrozole, which are used to treat the breast cancer. The N4 nitrogen of the triazole ring interacts with the heme iron of the aromatase and the prevents binding from the steroid substrate. The aniline nitrogen of the aminoglutamate serves as the same purpose. Formastane acts as a suicide substrate that permanently inactivates the aromatase and more selective in its action than aminoglutathione. So, so there are two types of these are the type 1 and type 2. Type 1 are the steroidal inhibitor, type 2 are the non-steroidal. First generation type 1 are none, but the type 2 are the aminoglutathione. Second generation, first type 1 is the formastane, and the third generation type 1 is the exmastane, and one is the second generation type 2 is the fedrozole and roctinamide, and the third generation is the anastrozole, letrozole, and borozole. So this is the reaction which is catalyzed by the aromatase. This is the aldosterone, testosterone, and this is the estrone or estradiol. Okay, this hydroxylation step is done by aromatase, and these are the structures of the competitive inhibitors of aromatase. This is the structure of the aminoglutamate. This is the structure of the anastrozole, and this is the structure of the letrozole. Okay. This is all about the aromatase inhibitor. Now let me go to the next problem. Problem number 70 says kinase inhibitors must bind to the dash region of enzyme. Your options are number A hinge, number B N lobe, number C C lobe, and number D DFG region. Which of the following will be the correct answer of problem number 17? Kinase inhibitors must bind to dash region of enzyme. Hinge region, N lobe, C lobe, or DFG region. Let me know. Kinase inhibitors must bind to dash region of enzyme. Your options are number A hinge, number B N lobe, number C C lobe, and number D DFG region. Which of the following will be the correct answer? Kinase inhibitors must bind to dash region of the enzyme. Which will be the correct answer? Hinge region, N lobe, C lobe, or DFG region? Let me check the chat box. Okay, number D. I have received one answer which is saying number D is the correct one. Let me check if number D is the correct answer or not. So the correct answer will be number A, hinge region. Okay, kinase inhibitors must bind to the hinge region of the particular enzyme. So this is first, let me just show you the structure of a kinase enzyme. So basically in kinase enzyme we have major three part. One is the N region, N lobe, which is this one, which is basically have the beta pleated sheet, and alpha helix are predominant in this position, and these are the C lobe. So in N lobe, this is the C lobe, and the joining of the N lobe and C lobe is done by the hinge region. Hinge region is the joining region of the N lobe and C lobe. The first amino acid residue of this hinge region is known as the gatekeeper residue, and also here we have a loop now where there are three amino acid DFG means are together. Okay, so these are now known as the DFG loop. So the kinase inhibitor, they must bind to the hinge region. So hinge region of the protein kinase is a short, flexible polypeptide segment that acts as a structural and functional bridge between the N-terminal lobe or N-lobe and C-terminal C-lobe of the catalytic domain. It is a critical component of the ATP binding pocket, providing the anchor points with the adenine ring to the ATP. The location, it forms a deep cleft and cleft-like interface between two main lobes of the kinase sequence. It typically consists of about three amino acids located immediately downstream from a key residue known as the gatekeeper. Nomenclature, these residues are often designed as the GK plus 1, GK plus 2, GK plus 3 based on their position relative to the gatekeeper. Conformation in many kinases, these residues adopt the extended beta sheet conformation, exposing their backbone amide and carbonyl groups to the solvent. Biological function, ATP anchoring. The backbone of the hinge region forms conserved hydrogen bonds with the adenine base of the ATP. Now, specifically, the carbonyl of GK plus 1 and the peptide amide on GK plus 3 typically serve as a hydrogen bond acceptor donor respectively. Lobe movement, as a flexible linker, it allows two lobes to move relative to one another in the hinge motion, and it is necessary for the kinase to transition between the active and inactive state during the catalytic cycle. The selectivity filter, the gatekeeper residue at the start of the hinge, act as a molecular filter that determines the size and shape of the hydrophobic pocket and bind to the ATP binding site. Importance in the drug design, the hinge region is primary target for the vast majority of the FDA-approved kinase inhibitor. ATP mimetic type 4 inhibitor compete with the ATP by forming 1 to 3 hydrogen bonds with the hinge backbone. Selectivity challenges because of the hinge backbone interaction are highly conserved across the human genome. It is difficulty with drug design, target only one specific kinase without hitting others. Inhibitor binding mode, researchers have identified that up to 15 distinct type of hydrogen bond binding modes between various inhibitors and the hinge region, which are used to guide the rational drug design and scaffold, hoping to create more selective drugs. Okay, now I will go to the next problem. Problem number 18 says, which of the following is not a PARP inhibitor? Your options are number A, rucaparib, number B, niraparib, number C, lapatinib, and number D, olaparib. Which of the following is not an PARP inhibitor? Number A, rucaparib, number B, niraparib, number C, lapatinib, and number D, olaparib. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 18. After a minute, I will check the answers. Meanwhile, let me know A, B, C, or D, which of the following will be the correct answer. Which of the following is not a PARP inhibitor? Rucaparib, niraparib, lapatinib, or olaparib? Let me know A, B, C, or D, which of the following will be the correct answer for problem number 18. Let me check the chat box. Okay, I have received an answer which is saying that number C is the correct answer. Okay, let us find out if number C is the correct answer or not. So the correct answer will be very good. Number C, lapatinib. It is not a PARP inhibitor. It is a kinase inhibitor. Okay. So, a number of research groups are investigating poly-ADP ribose polymerase or PARP inhibitors as the potential anti-cancer agent. Although none of the reached to the market to the day, the enzyme repairs the single-strand breaks in the DNA, and so the inhibition can eventually result in the double-strand breaks in the DNA and the cell death. Now, some tumor cells that are likely to more susceptible to PARP inhibitor than normal cells. So basically, this is the some this reference is old. So these four actually have reached to the market after completing the successfully all the clinical trials, and these four are the FDA approved PARP inhibitor which are present in the market nowadays. Okay, so olaparib, rucaparib, niraparib, and talazoparib. These four are the FDA approved PARP inhibitor. These only four molecules are approved till now actually. Okay. So basically, In recent years, PARP or poly-ADP ribose polymerase have gained significant attention in the cancer treatment. Studies indicate that PARP play a crucial role in DNA repair, maintaining genomic stability, regulating cell activity, and responding to the DNA damage. Consequently, with the discovery and exploration of PARP inhibitors are emerged in the cancer treatment. These inhibitors are now used in clinical application for tumor therapy, particularly for BRCA mutation-related cancers. A series of PARP inhibitors have been developed for the cancer treatment, with several approved for clinical use in the cancer therapy. These inhibitors disrupt the DNA repair by inhibiting PARP enzyme activity, leading to the cancer cell death, thus offering potential for the tumor therapy. We summarize the biological mechanism of the PARP inhibitor, focusing on the BRCA synthetic lethality, recent research development, clinical application, improvement of the existing inhibitor, and discussing their potential mechanisms and challenges in the cancer therapy. So these are the PARP targeted agents, single target therapies. Rucaparib, niraparib, talazoparib, they are approved drugs for the olaparib is the first in class inhibitor, and these are the dual targeted therapy with PARP with other inhibitors. Okay, the promising synergistic combination of PARP with other anti-tumor agents, they generate new anti-tumor agents which are very efficacious. Okay. Now let me go to the next problem. Problem number 19 says anti-ulcer drug omeprazole is number A reversible proton pump inhibitor, number B antagonist of H plus K plus ATPase, number C agonist of H plus K plus ATPase, and number D irreversible proton pump inhibitor. Anti-ulcer drug omeprazole is which of the following? Number A reversible proton pump inhibitor, number B antagonist of H plus K plus ATPase, number C agonist of the H plus K plus ATPase, and number D irreversible proton pump inhibitor. Which of the following will be the correct answer of problem number 19? Anti-ulcer drug omeprazole is which of the following? Again, I will wait for a minute, then I will check the chat box. Okay. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 19. Anti-ulcer drug omeprazole is which of the following? Number A, reversible proton pump inhibitor. Number B, antagonist of H plus K plus ATPase. Number C, agonist of H plus K plus ATPase. And number D, irreversible proton pump inhibitor. I will check the chat box after a minute. Meanwhile, let me know A, B, C, or D, which of the following will be the correct answer. Let me check the chat box. Let's see if there is any answer for our problem number 19. Okay, number D is the answer that I have received. Let me check if number D is the correct answer or not. So the correct answer is yes, very good. Number D, it is a irreversible proton pump inhibitor. This is the structure of omeprazole. So the proton pump is the only present in the canalicular membranes of the peritoneal cells and the crucial from the mechanism by which hydrochloric acid is released into the stomach. It is called as an H plus K plus ATPase because it pumps the protons out of the cell into the canaliculus. At the same time, it pumps the potassium ions back in. Energy is required for this process as both of the protons and the potassium ions are being moved against their concentration gradient. In fact, the ratio of the protons inside the cell and the protons in the canaliculus is 1 is to 10 to the power 6. The energy required to carry out this exchange obtained from the hydrolysis of ATP, hence the term ATPase. The pump is not responsible for the efflux of the chloride ion. This departs the cell through the separate chloride ion channel. This outflow closely matches to the efflux of the protons and such that the chloride ion is released for every proton that is pumped out. As a result, hydrochloric acid is formed into the canaliculus and rather than inside the perioral cells. As each chloride ion departs the cell, it is accompanied by the potassium ion which flows through the zone channel. No energy is required for this outflow because of the potassium ion is flowing down to the concentration gradient. The potassium ion acts as a counter ion in the chloride ion, and once it is canalicrous, it pumped back to the cell by proton pump as described previously. So this is the proton pump, the role of proton pump in secreting the hydrochloride acid. Okay. So there are four proton pump inhibitor in the clinical use: the omeprazole, lansoprazole, pantoprazole, and rabeprazole. The S-enantiomer of omeprazole has been approved for recently. All the proton pump inhibitor have a pyridinyl methyl sulfonate. So this is the pyridinyl methyl sulfonide benzimidazole skeleton as the prodrug. Since they have activated, they to reach the acidic canalicular peritoneal cell. Once activated, they bind irreversibly to the exposed cysteine residues to the proton pump and block the pump, preventing the further release of the hydrochloric acid. Now, this is a big strategic advantage in the inhibiting the proton pump rather than blocking the histamine and cholinergic receptor. For example, H2 antagonists block the histamine receptors and block stimulatory effects of the histamine, but it does not block the receptors for acetylcholine or gastrin. So it is still possible for the peritoneal cell to be activated toward the secretion. The proton pump is downstream of this target and operates the final stage of hydrochloric acid release. Blocking it presents the release of the hydrochloric acid regardless what mechanisms are involved and in stimulating the hydrochloric acid secretion. So this is about the proton pump inhibitor. Now the last question of today's session. It says which one of the following activities occurs following the stimulation of the H2 receptor. Your options are number A vasodilation, number B uterine contraction, number C bronchial smooth muscle contraction, and number D enhanced secretion of hydrochloric acid in stomach. Which of the following activities occurs following the stimulation of H2 receptor? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of the hydrochloric acid in stomach. Which one of the following activities occurs following the stimulation of H2 receptor? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of hydrochloric acid in stomach. Which one of the following activities occurs following the stimulation of H2 receptors? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of hydrochloric acid in stomach. Let me check the chat box. Okay, number C, and number D. So I have received number C and number D, two different options as the answer. Okay, let me check which of the following will be the correct answer. So the correct answer will be number D, enhanced secretion of hydrochloric acid in stomach. This activity occurs during following the stimulation of H2 receptors. So histamine exerts its effect through the four primary receptors. H1 and H2 are the most clinically significant. While H1 receptors are allergic targets found in the airways of the and the skin, H2 receptors are the digestive target of the primary located in the stomach. Comparison between the H1 and H2 receptor primary location. H1 receptors in the smooth muscle or than the vascular endothelium and brain, whereas the H2 receptors are present in stomach, the peripheral the cells and heart and vascular smooth muscle. Main function mediates allergic reaction, itching and wakefulness in case of the H1 receptor. H2 receptor regulates the gastric acid secretion and heart rate. Effect of activation H1 receptor bronchoconstriction, vasodilation and increased vascular permeability. H2 receptor stimulation of hydrochloric acid production in the stomach. Clinical blockers H1 blockers cetirizine, loratadine and diphenhydramine are the H1 blockers. H2 blockers the famotidine, ranitidine, cimetidine. Common uses allergic rhinitis, hives and motion sickness are the common uses of the H1 receptors, and H2 receptor acid sickness, the peptic ulcer and heartburn. So key difference of action respiratory versus digestive. H1 activation narrows the airways or bronchoconstriction and keep clear in the asthma and allergies, whereas the H2 activation primarily handles the burn by triggering the acid release in the digestion. Central nervous system H1 receptors at the brain help to maintain the alertness. This is the first generation H1 blockers like Benadryl cause the significant drowsiness as they cause the blood-brain barrier. So basically, the enhanced secretion of hydrochloric acid in stomach is the activity that occurs during the stimulation of H2 receptor, because H2 receptors are basically present in the stomach, and they stimulate the hydrochloric acid secretion of the stomach. That is main important role of this H2 receptors. Okay. So this was the last question of today's class. So today what we had discussed majorly, we have discussed three type of therapeutic agents. Broadly, we have discussed about the antiviral agents, various antiviral agents, the mechanism of actions. And then we have discussed about the anti-cancer agents majorly. So anti-cancer agents also we have discussed when we were discussing about the nucleic acids as the drug target. That class is also important regarding the anti-cancer agent, because many anti-cancer therapeutics, their mechanism of actions we have discussed already in that particular class. Okay, and then we discussed about the anti-ulcer agents a little bit in today's class. So this was, let me share my email ID. If you have any doubt or any query regarding today's class or regarding any of our previous classes, okay. Right. Let me first share my email ID. So this is my email ID, as we are almost at the end of our course. So if you have any doubt or any query regarding any course, any topic, okay, let me know via email. We will be having our last class on next Monday from 6 to 8 p.m. And then after on the day after that, means the on Tuesday, we will be having a summary class. The exact time and the schedule will be communicated to you later. But if you have any query or any doubt, please let me know through the email. Okay, so that I can, I can prepare accordingly, and I can answer those queries in the particular summary session. Okay, so that's it. So today's class, if there is no query, I will close the session. I have already mentioned where to find all the material. You have to go to the SWAM portal. On the announcement page at the end, you will find the problem solving session. If you click on that problem solving session recording, you will be redirected to a page which looks like this. This has all the class recording and the Google Drive link which contains the presentation. Okay. So if there is no doubt or query, I will close the session for today. Thank you all for joining, and thank you all for answering. We will meet on next Monday from 6 p.m. Okay. Thank you all. I will close the session now.